Om Shri Sairam. Welcome to the evening session of the Indo-US conference 2. So we have been waiting long for this evening sessions, and we are keen to listen to the world-renowned speakers. Over to Dr. Khanna for further proceedings for this evening session. Dr. Khanna.
Good evening and welcome to the evening session. For the first keynote address this evening, we have Dr. Olcay Akman on the parameter estimation in compartmental models under frailty. Dr. Olcay Akman is a professor of mathematics at Illinois State University in Normal, Illinois. He is the director of the Intercollegiate Biomathematics Alliance, a consortium of universities collaborating and sharing resources in the pursuit of scholarships, teaching, and advanced research and development. Dr. Olcay Akman is currently editor-in-chief of the journal Letters in Biomathematics and a journal for undergraduate research, Spora. His research interests include compute-intensive modeling using evolutionary computing and machine learning-based methods. Over to Dr. Olcay Akman to deliver his keynote address.
All right. Thank you very much for the introduction, and it was an honor to be invited to this. And yesterday I was at the opening, and I admire all of your efforts for putting together this wonderful conference, and it's really great pleasure to be a part of it. Thank you. So let me. So the parameter estimation in compartmental models under frailty has been not a new topic, but with COVID, the focus on frailty, focus on patients with underlying conditions being affected from COVID, frailty models took the front seat in modeling. So I'm going to share really very basics of frailty modeling as an option for the future researchers of infectious diseases to implement this in their modeling approach. So here's an oversimplified, perhaps, an introduction. We here have a very basic SIR model, and when we look at typical SIR models, generally the parameters that dictate from transitioning from one compartment to another are generally fixed. So by generally fixed, what we mean is if you are in the susceptible compartment, your basically chances or probability of being infected, in other words, your chances of being transitioned to the infected compartment is considered to be same as everybody else in that compartment. However, as in most infectious diseases, especially in COVID, this is not the case. We already know 65 plus portion of the population has different rate of suffering from COVID than, let's say, 18-year-old healthy portion of the population. So since the population itself is segmented, the reproductive rate of disease, the incubation rate, recovery rate, mortality rate, all vary. And frailty, by the way, is not only a physiological condition. You don't have to be let's say 65 plus, and a diabetic and an obese person to be negative, to be adversely impacted from a disease. You may be at the same age as another member of the population, but you may live in a region of the country where you don't have much access to quality healthcare and compared to other members, or you may live in a part of the culture that has a adverse dietary trends than let's say someplace else where the healthy diet is followed. So for many studies, the population itself is divided based on these physiological, economical portions where the factors affect these portions differently. So for instance, in STDs, the disease distinguishes between males and females, partitioning an SIL model into different states. So the moral of the story basically is individuals are not similar, and the persistent differences among individuals, and some of which could be due to the environments in which they live, or their susceptibility, or their socioeconomic conditions may actually impact the acquisition of the disease. So frailty in its simplest form is an unobserved random factor that modifies multiplicatively the hazard function of an individual or a group or a cluster of individuals. And with the concept of frailty, we provide a suitable way to introduce random effects in models to account for this unobserved heterogeneity. In basic, simple sense, if you were a pure statistician, you would call this factor covariate. But we are going to, in compartmental models, in ODE models, we are going to treat them differently, but they serve the same purpose. So how does heterogeneity influence the dynamics and intensity of infections? Individual differences in susceptibility or immunity to an event, and different patients possess different frailties, and patients more frail or more prone tend to have the event earlier than those who are less frail. When we say an event here, we are talking about an infection, or we are talking about a change that moves you from one compartment to the other compartment. So let's define a function f as the intensity for the occurrence of an event, for an infection, for instance, for an individual, individual I at age A at time t. And the patient's frailty is quantified with W. So if W is 1, where our function becomes then the occurrence of the event at age A at time 1 with a standard individual. If the W is 2, which means that that patient is twice as susceptible to the event as the standard individual, and obviously if it is less than 1, the individual is less susceptible. So in other words, we can think of the frailty as a multiplicative effect where the more frail you are, the more chances that you will be subject to the event. So in a typical group, since I started using the example as Scir, SEIR model, I'm going to list perhaps the components of the SCAR model. So let's say X, the differential X be the proportion of individuals in the susceptible group. H is the proportion in the exposed group, and Y is the infected group, and Z is the recovered or the removed group. And in a typical publication, we would use these as these typical flowchart model where we move from box S to box E, and then the infected group, notice here, is divided into subgroups, and these subgroups I1, I2, and I3 are the compartments that contain patients with different frailties. So if XIJW at HA indicates the proportion of susceptible individuals, these individuals acquire the infection from all from the infective subclass through force of infection. Let's call this force of infection WI, which involves the exposed class H. So lambda I is here the key point. Then the individuals that develop the disease with some rate sigma, and then we just keep moving the patients from group to group. What's the force of infection? In force of infection, if you were again a pure statistician, you would call that failure rate or hazard rate. That's simply per capita rate of acquisition of infection. So Fi is the observable variable. For instance, we observe that from well-known field studies. Unfortunately, that in COVID-19, that took a while for us to gather enough baseline information to have a value on this F. So we then calculate F given the population is heterogeneous. Since I have a limited time, I'm going to perhaps pass through these rather quick and like to show a simulation with this. So for sake of simplicity, I am going to discuss this based on a group with only two subgroups. Again, this is oversimplified, but I think it will convey the message properly. Suppose in our population we have two groups of susceptibles, say old people and young people, and our assumption is old people have higher chance of disease acquisition. So if that's the case, our model will accommodate these force of infection for different groups with parameters lambda 1 and lambda 2. The rest are your typical ODE model. Basically, the individuals in group 1 are susceptible with some coefficient, and then they move to the next group. And then similarly, the individuals in group 2 have their own susceptibility rate, lambda, and the argument is if you're in the old group, your lambda is bigger than 1, greater than 1. If you're in the young group, let's say lambda is less than 1, and the rest is your standard introduction to ODE modeling details. So here, sigma is the average incubation period, gamma is the infectiousness period, and then mu is the age-dependent mortality rate. So by solving these, we can basically, we are doing all of these to get the two things. One is the populational force of infection. And the populational force of infection can be defined as the weighted average of these subgroups. The difference in here is currently the typical COVID model, when we, for instance, say, oh, for 65 plus people, the infection rate is, or the mortality rate is 8%, but for 18-year-olds, the mortality rate is 2%. When we say that 8%, we are putting all 65-year-olds together into a group without counting, without quantifying the fact that some of them could be diabetic, some of them could have cardiac conditions, some of them could be obese. We don't really look at that. This populational force of infection represented as a weighted average actually addresses this problem. So we now then we focus on the force of infection. As I said, one of the underlying assumptions of the model is this frailty is implemented in the model as a multiplicative coefficient. Of course, it doesn't have to be, and that can easily be worked out in other forms. And so here we'll have the two components, the basic intensity hazard and the mixing variable. Here, I think, is, believe it or not, in this entire presentation, this is my favorite slide. The reason this is my favorite slide is we can look at the population as in terms of time into two subgroups. Again, this is my oversimplified example. So one subgroup is the high-risk subpopulation, and the other subgroup is the low-risk subpopulation. So what happens is, as time progresses, the high-risk subpopulation members will start dying. So it's because they have a higher mortality rate. So as their portion in the population will start getting lower and lower, the estimated hazard rate over the entire population will appear to be decreasing, because now population is slowly being dominated by the low risk members, although the estimated hazard rate is actually still really the same estimated hazard rate. It's just appearing to be decreasing because the high risk population members are decreasing. So that's basically pretty much what this slide says. Failure rate in the surviving population will fall over time because the portion of the high risk and low risk subpopulations in the population, in the overall population, will change. So this is the kind of heterogeneity that is not taken into an account when we report disease statistics. So here's a very quick example. We have 80 subjects, and at time t initially, we look at the correspondence to the date of diagnosis, and then analysis time is years until death, or a patient leaves the study before they die, at which time we don't have their lifetime, but we know the point until which they survived. Again, in this case, the covariates or the frailties are smoking status and obesity, basically, or weekly calories from fat in their diet. Okay, so this is a quick example. These are the ID numbers. And then if the patient is smoker, the column will represent this as 1, otherwise it's 0, and this is the normalized coefficient, normalized data for the diet. Now, here, ignore everything else, just look at here. So the hazard ratio corresponding to smoking and the calories obtained from fat in their diet are basically the smoking contributes about 5 units in terms of the variable, and the fat diet contributes about 7. Now, if I omit these failure variables, if I omit these covariates, which are basically unexplained heterogeneity, then this is what happens. First of all, we look at a different frailty model, and look, since I omitted those 2 variables, what happened is we are only looking at age and smoking. So it looks like smoking puts you in risk little over or little under 3 times, and then your age is also a concern in here. And let's change the model from gamma to some other distribution, and notice that the age and smoking rate stayed about the same, because in this case, with the correct parameter estimation, inverse Gaussian and gamma shapes are similar, so it looks like our model is stable. But now let's introduce the frailty, and in this case, what happened is the impact of diet fat now started getting into the picture, and now it's affecting the other variables as well. So according to this, the older you get, your hazard rate increases. However, your smoking and diet combined together is more of a concern for you, for your mortality, than the age itself. And so since I have only just few minutes left, the question we would like to rate is, when we build a model, we generally build a model and put our faith into that form of the model and try to make it optimal by adjusting the parameters. What we want to, the message that I like to convey in here is actually model itself need to be flexible in order to quantify, in order to accommodate to hidden heterogeneities of the model. So the question that I like to raise is, do I want an observation level frailty, or do I want to impose a grouping constraint on the frailties of the patients? So here we know which model is more appropriate, but in practice we need to ask this fundamental question: how does the choice of frailty distribution affect the reproduction number? So where the reproduction number here is given in as a function of transition parameters. And here again, beta is the age-independent effective contract rate. Okay, so in conclusions, I hope I was able to convey the message that populations are not a group of identical individuals. There is heterogeneity, and heterogeneity is an important, very important variable to be taken into account for especially the interpretation of epidemic data. And it can help in the systematic understanding of the population's makeup and its effect on the spread of infection. So for our young researchers who would like to work on this abundant opportunity that basically came to our labs with COVID-19 or similar variations of COVID data, here is some couple possible future directions. we may need to derive a set of equations which basically define the reproductive number for different frailty behavior. My example was oversimplified with just two subgroups. And also, there is a great, I think, potential for the estimation of the effect of heterogeneity on the spread of infections and on its effect on R0. So with that, I would like to thank the organizers for giving me the chance, and I believe I have just a couple of minutes to answer any questions.
Yeah. Thank you, Professor Akman. So I have a quick question. So this Frailt about the Frailt and the distribution, are there any kind of things related to COVID? Because I was seeing your talk was based on diabetes and a few more the behavior such as smoking and some other patterns. So would be little interested the same scenario where suppose if we try to look for COVID, so how can these parameters or the distribution that can help in understanding the parameters, especially with reference to COVID, what should we look at?
Right. So actually that flowchart model I showed was from a paper that we published in with one of my graduate students in the journal Spora. We basically studied COVID, particularly COVID, and the frailty component that we studied was basically poverty and access to healthcare. So the choice of distribution, as your question focuses on, comes into the picture that frailty parameter is stochastic. We don't know, and it has so when I say, for instance, oh, if you're old, you're twice as likely, and if you're young, you're half as likely. That actually is, of course, some deterministic blind number. The idea in there is, can we actually put a distribution over the frailty parameter and then implement that as a random variable in the model and find the optimal frailty for a particular subpopulation? The distributions in my example that I used, gamma and inverse Gaussian, are distributions because those are the ones with positive support, positive domain. I believe determining the optimal frailty parameter is not any different than determining the optimal model, where instead of, let's say, suppose, for instance, you have an SIR model with parameters alpha, beta, and mu. Instead, now you don't have alpha, but you have lambda times alpha. And let's call it alpha star. Optimize alpha star.
Okay. Thank you, Professor Okman. Dr. Anuj?
Yeah, well, the time is very limited, so I would just briefly says, I mean, most of you know Dr. Akman, but he has been very kind, and you can see the logo on the background of the conference. There's a logo of Center for Collaborative Studies and Mathematical Biology. Its executive director is Dr. Akman. And so thank you, Dr. Akman, for accepting our invitation and actually supporting this conference. It has been going on with some level because of your help. So thank you very much. And I will not take much time. I know, I mean, I can communicate with you at a personal level.
Thank you. And at the risk of taking 15 more seconds from our next speaker, for everybody else who are involved in this conference, I like to say one thing. I met Anuj in 2018 during a biomathematics ecology education and research conference. I can say my career took a different turn after I met with him. He's a treasure. He's a gem. So if you can get hold of him to have a chance to work with him, I would very highly recommend it. He's my idol. Thank you very much.
Thank you, Ojay. One more thing, actually, very quickly. Dr. Akman is also running beer conference, which is just now coming up in November. Maybe I will circulate it to the all participants, the link of it. So
that's right. All right. Thank you.
Thank you, Professor Akman. Thank you, Dr. Anuj. For our next keynote address this evening, we have Professor Guo Wei on forecasting vaccine breakthrough SARS-CoV-2 variants. Professor Guo Wei is a Michigan State University Foundation professor. He received his PhD degree from the University of British Columbia in 1996, with a postdoctoral fellowship awarded by the Natural Sciences and Engineering Research Council of Canada. He did research work at the University of Houston, where he became a research assistant professor of chemistry in 1997. In 1998, he joined the faculty of the Department of Computational Science at the National University of Singapore and was promoted to associate professor in 2001. In 2002, he relocated to Michigan State University, where he is a professor of mathematics, professor of electrical and computer engineering, and professor of biochemistry and molecular biology. Over years, Dr. Wei has accumulated research experience in many disciplines, including mathematics, physics, chemistry, biology, computer science, and engineering. His current research interests include mathematical molecular bioscience and biophysics, drug design, deep learning, and bioinformatics. Dr. Wei has served extensively in a wide variety of national and international panels, committees, and journal editorships. Without much delay, now I invite Professor Guo Wei to make his keynote address. Over to Dr. Wei.
Can you see my screen?
Yes, sir.
Yeah. Okay, thank you very much for the introduction. It's my honor to present my talk in this conference. Yeah, also thank you very much for the kind introduction. I'm in the mathematics, but I have a courtesy appointments in the two other departments. So my talk is focused on the correlating. So correlating, as you know, this is a horrible disease, and it has so many, 240 cases and almost close to 5 million casualties at this point. If you look at Google Scholar, you see for the two things you search over there, like a correlating and SARS-CoV-2, it's something about almost is something over there near 400,000 articles, all kinds of different material over there. So the question is how will correlating unfold in the future? This is both from an academic point and also from a pandemic point from human health. So I would like to give a little bit of introduction to from a basic knowledge we have about the disease. This is a virus over there. It has a spike protein and also immunoprotein, membrane protein, and also there's a M protein over there. Is a protein is associated with the virus genome. The messenger RNA. So that's the major proteins are those ones. And then you get into the system. This is the human cell over here. So the blue part is the human cell. Get into the cell through the help of two proteins here. One particular one is ACE2. That is goes through with the spike protein, right? With the S. So this is spike protein binding with ACE2, and this is a channel such that the virus get inside our cell. After they get inside the cell, it's just release the viral material and then produce the proteins and different proteins over there, and also replication, so and the transcription and the translation get back into those four proteins over there, right? So this and then they become a three-dimensional protein, and then they assemble the virus again and get back the cell surface, and then to infect other cell. See, this is a cell cycle. So you realize this is a really complicated process, right? I mean, it's so complicated, and each different place also has spent like 10 or 20 years to study. And what is exactly the process over there? So, but from mathematical point of view, I mean, we want to see things from the mathematical point of view. Try to simplify what is the major important things over there. What is the most important things in this whole process? You may say everything is important, right? This is the whole thing put together, make it a horrible virus. But if you form the virus transmission and for the virus evolution from that point of view, the most important thing from my point of view is here. So this is a point, is a point of contact, and this is a spike protein interaction with ACE2. This is crucial. I mean, without this interaction, the virus will not affect us, right? The virus might not affect, for example, some other things, mosquito or something like that. It does affect a lot of different animal and human. And because we have ACE2, but if you don't have ACE2, I mean, they won't affect us at all. And this is a crucial point. And anything around this interaction, possible structure change, possible mutation, whatever, is going to determine the dynamics of the whole process. That's the point. So you see, from a mutation point of view, and the mutation happened for the virus everywhere. This is a virus, you see, go from N protein, NS1, NSP2. So it's non-structured protein 1, non-structured protein 2. And here the final ones, like some N protein and also OIF, so open reading frame. Some other things goes to 10. This is the endpoint. A total of the 29,000 of nucleotides. This is the whole genome, right? And this each point here, what it means, each point here is one mutation. And how many mutations we have? I mean, the mutation is horribly a lot, because this is a natural log scale for the frequency. And also this natural log scale goes to 16. That's huge. So that means some of this mutation happened like almost 2 million times, right? Something like that. But a unique mutation, we only have something 28,000 unique mutation. But it's still too many, right? Because our genome size is 29,000. So that's based on average, each place there's one mutation, right? Each nucleotide. This is horrible many mutation. How can we make sense out of this mutation? So that's the question. How can we make sense out of this whole thing, right? So this is what governs the mutation, governs the transmission and the evolution. Mutation means evolution for the virus. What governs the evolution of the virus? This is a question we try to answer, come out of this such a horrible complexity. So if we really look at the mechanism of a mutation, competing different mechanism mutations, right? There's the first thing is a random genetic shift. I mean, as it can happen in all different places, like replication, transcription, translation. I mean, those things I show you in my earlier slide. And also it has a recombination. So it has like a viral pool reading, because virus is quite smart. This virus is unlike a flu virus or HIV. They don't do any pool reading, but this virus is a big virus and they have a mechanism pool reading, so they can correct the mutations over there. and also the host genome editing. So when virus get into our body, our host try to do some type of editing of the genetic sequence of the virus and try to destroy the virus. So our host immune system has such ability. And also recombination, that means the virus recombination, the same as this one, but happen at a different scale. I get into that. And also natural selection. So if you summarize everything here, these are the molecular scale, okay, events over there. And these ones are the organelle scale. So recombination here means the genetic recombination combined with our human body recombination, things like that. That does happen where it's not very often, but it did happen, okay? And also there's a population scale. So this is so you see we have a multi-scale, molecular scale, organelle scale, population scale, natural selection. I will explain what is natural selection just in a few seconds over there. So if you see here, this is what we try to do. Really, this is the whole process. What do we do? We have a program we established in the past almost 2 years now. We have a combined genome and math and AI experimental data for the model of protein-protein binding affinity change follow mutation. What it means, you see, this is our patient. From the patient, we extract the virus. From the virus, we analyze their sequencing. So we put the data into the guide side. From there, we take over from here. We take over, so we do genome typing. So basically, we use the computer to try to compare with the earlier genome reported in Wuhan. That's in Wuhan, January 2020. So we compare the current the genome, the virus genome from a patient, and compare with them. Actually, at this point, we have more than 2 million of that, and we found something like 28,000 of mutation over there. And from those 28,000 mutations, I want to focus on something so-called receptor binding domain. What is a receptor binding domain? Receptor binding domain is here. You have ACE2, right? That's a human ACE2, right? Receptor. And this is a virus spike protein. The spike protein, the tip of the spike protein is called a receptor binding domain. It's a RBD. So this is the point. It's the most important point. We focus everything on this. So this is about roughly about 200 nucleotides, okay? Amino acid. There's about 200 amino acid and this human receptor, and their interaction, everything mutation happen here is very important, right? So this is something we try to look at, this interaction, how each of those mutations, just 700 mutations, going to affect the binding between the host ACE2 and the virus spike protein. That's going to affect the infectivity. And also, I can replace this ACE2 with antibody. Antibody is a material generated from our immune system. And that antibody also tried to bind to this spike protein RBD, and such that this RBD is no longer be able to bind it with ACE2. That's the principle. We have a vaccine, right? The vaccine, most vaccine is tried to generate antibody such that it binds to this spike protein, and the ACE2 cannot bind. And at this point, we actually collect something like 130 antibodies from different patients. We have their structure, we have a detailed information. So from there, what do we do? We use topological data analysis. So it's a persistent homology, topology, things like that. We analyze user mathematics. And from there, we go to deep learning, like a convolution deep learning, like a convolution, pooling, drop, and drop out, things like that. So we use basically deeper neural network, okay? And from there, we get the like a binding free energy change. That's the energy change, how the energy change affected by individual mutation. So between this ACE2 and the spike protein RBD. Otherwise, also between the spike protein RBD and the antibody. Okay, at this point, it can be ACE2 or it can be antibody. And in this way, we can analyze the infectivity change induced by mutation. So that's the idea. That's basically all we do. So I just wanted to do a tiny bit since like a few seconds, 10 seconds. So basically, we want to start a protein with something so-called simplicial geometry. We talk about a simplicial complex. So it's just dots and lines and triangle, whatever, so on and so forth. From there, we have a chain and form a chain group. And from chain group, we use a boundary operator to generate some other things like a circular group and a boundary group, things like that. I mean, that's a lot of basically what we want. We really want the homology group. So this is how, after we have a homology group, the dimension of the homology group corresponding to the topological invariant, so-called Betti number. That's the stuff we use. We use that Betti number in the topological barcode I showed you a moment ago, right? And this whole thing also can be make it more powerful user filtration. And we end up with not just algebra topology, we have a persistent homology. I don't want to get into too much detail of this math. So I just want to show some pictures, make it more interesting to see. This is all the color like this one, This is just RBD, the spike protein virus. And those are this is ACE2 binds to spike protein. And then it's competed with something like antibody, like a BD620 line. This is one particular antibody. And also CC12.3, this is the other antibody. You see, they competed with this ACE2. This is ACE2, right? And so this is how, after they bind to this place, ACE2 can no longer bind to the spike protein. That's the whole working principle for the vaccines and antibody drug. And we have 130 of this type of stuff. I just show you like a 60 or something, 46, right? So what we did, the things we did is we have one paper published, actually the time was published in the July of 2020. So it's last year, July. So and in this paper, we did quite a lot of things. We just use algebra topology, you can see there. But the one thing, we did three important things in this paper. The one thing I wanted to say we are very proud about this paper is we predicted the SARS-CoV-2 variants. So before we report any variant, so it's like a half a year before anybody reported any variant. So we actually predict variant position at the receptor binding domain, RBD. This word happened many times, right? So the residue, there are actually almost 200 residues over there. So means amino acid. So we actually point out 452 and 501, those two important. We certainly not only two, right? We point out 452 and together with three others. But this is come out from 200 residues. We point out this 5, but after like a half a year later, we realize these two are so important. How important is, you see, now we have a lot of different variants over there, right? We have a key variants, most important variants, and we have studied earlier like alpha variants. That's the one initially identified in the UK, and the beta, that's initially identified in the South Africa, and gamma, this is Brazil, right? Identified over there. And then you look over there, alpha, beta, gamma, delta, and epsilon, theta, kappa, lambda, mu, all most of the important variants, they have mutations at 501 position, right? While otherwise it's 452 mutations, right? Position. So we, as I said before, we predicted this just like half a year before anybody announced anything. Alpha was identified in the last December, right? So something like that. So we actually used our mathematical method to identify most important mutations. Now is Galvin mutation in the all most important variants over there, right? And that's just one part of our work. Second part I would like to share with you is we actually discovered the mechanism of virus transmission and evolution. Why we claim that? So in the same paper I showed you before, published in the July of 2020, this is a copied over there, right? So we copied this figure and this is a paper. We said we hypothesized the natural selection favor those mutations that enhance the viral transmission. And if our predictions are correct, we don't know if they're correct or not at that point. We don't really know. We know now is really, really correct. It's also not only correct and also accurate. So if our predictions are correct, this is a theoretical, right? It's not experimental results. And the predicted infectivity strength mutation, we outpace predicted infectivity weak mutations over time. I mean, at that time, we don't know what over time means, but a few months later, everything approved just to confirm what we said before. So you see, these ones, the red ones, you see, represents mutations that strengthens the infectivity. So those ones, their frequency is high. At that point, we have less than 100 mutation on RBD. And the frequency is only as high as 50, right? Something like 51, 52. Really, really not very high. What is the frequency now? The frequency now is like a 1 million, right? Now, at that time, it's really, really early stage, only 50. And then we spark a few red ones, you see? And they outpace the blue ones. The blue ones are the mutations. So each line is a mutation, and when it happens, as we show here, right? This is the time. So each line is a mutation, and the blue ones are the ones they make the binding, the ACE2 and the RBD binding weaken, right? So that's the ones they decrease the infectivity. So from here we imply, okay, natural selection will be the major issue over there. So that's what we did at really, really. Just a long time before anything figured out, because I mentioned you before, there are a lot of different competing mechanisms. We pick up this one, natural selection. And so certainly this thing is a genius Darwin. He forecasts everything if you have a species composition, and this natural selection is always there, right? And this is how you can see, like in the July of this year, and those mutations. You see the frequency goes to like a natural log scale is 12 or 13 or something like that. So that means something like tens of thousands of times. And this is the one is 501 and 452. Those are the ones that are dominated mutations over there, right? And also particularly those two, they are in the dominated variants. So this was only to the April, okay? So you can see the pattern is so, and if you look at it different way, and this is we look at it from the time. So this is a lateral log frequency. So mutation we observed. Look at the top 100 mutation, right? And you see the blue ones are the ones make the virus more infectious, and the red ones make the virus less infectious, right? So the red ones initially still can occupy a few places. The frequency is a lot high, right? This is just 1.5 of a lateral 10 scale. So that means something like a highest one is only less than 1,000, right? Something like that. If it's 2, that would be 10. That would be 1,000. It's 100. Yeah. So this is less than 100. Sorry. Yeah. Yeah. You can see, but this is a July. This is just the April last year, right? And now if you see after something here and here is in the February of this year. So all the top 100 mutations, they are the one. Each one is one mutation each bar. They are the ones they make the virus more infectious. So I mean that's the pattern you can see, and you can see from a different way. This is the top 100 mutation, right? And then look at the binding free energy change. This is the predicted ones, okay? So you can see the binding free energy change, they're positive, right? So all of them are positive. A few of them negative, but doesn't matter. The reason is they are all bigger than the average energy, right? If I look at all the energy, the binding free energy, or for this is like a lot of all possible RBD mutations, and you look at the binding free energy change, the average is negative 0.28 kilocal per mole. but all of these ones, top 100, are bigger than that. So if the things happen randomly, what is going to happen? That means each mutation has a half possibility going up, half probability below the average, and half probability above the average. In that case, if I have 100 mutations, the possibility for all of this 100 mutation to be above the average is going to be 2 to the 100, right? So that's given me a number. It's huge. It's 1.2 non-minimum. So that means this does not happen by chance, right? If it's by chance, just a 50-50 possibility. So this is just one configuration out of this many configuration. So that means that really, this is a natural selection. This is a deadly pool. This is a pool to the something. So it's to the accuracy of 1.2 times 10 to the 30. So that's also, again, says the natural selection is correct. Yeah. But also in that paper, we did one more thing. This is the last thing I wanted to say, report to you. I said they are in the receptor binding domain mutations, right? They are 3,686 possible mutations. Among those mutations, we put into 3 different categories. One is 1149 most likely mutation, and the other one is likely mutation and unlikely mutation. So at this point, all the mutation I show you, okay, it's 737. All the mutations happen over there. They all belongs to the most likely in that particular category. So that means our classification also is correct. So that's the 3 major things we did in that paper. And in the past 1 year, we published something like 20 papers on correlating. I just wanted to show you this one particular paper results we have over there. From here, now I wanted to talk a little bit about the antibody therapies, right? So, and they are actually, this is the older number. Now at this point, there are more than this many antibody in clinical trial. So this is how it works. This is our blood, right? It's a red blood cell, and there are something so-called B cell, and also we have a host cell. This is host cell has ACE2, and the B cell is produce antibody, right? This is the antibody, right? And this antibody, what this antibody does? What it does is try to neutralize the spike protein, right? And after the neutralization, I already said this many times. And this virus, the spike protein cannot just imagine area of this spike has an antibody attached to it, and it has no room to affect our human cell ACE2 any further. That's how the vaccine works. That's also how the antibody drug, if I design antibody drug, and that's how it works. So we design antibody drug such that it's going to neutralize this spike protein. So that's how it works. So this is our prediction. You see, this is the first one is our prediction of user DP mutation experiment. The DP mutation experiment is RBD binds to. This is a RBD binds to what? The RBD binds to antibody. This is CTC-445.2. This is a particular antibody. And this antibody, people actually tried to optimize this antibody by what? By mutate all of different residues, okay? So this is a different residues here. On each residue, they have so they mutated into 19, a total different amino acid, different type of amino acid, right? They just from each one, they mutated into all other 19 different choice. That's how, and then they look at the binding energy change after that, right? So, and in that way, they can actually systematically design optimum antibody. So this is the experimental result. This is our prediction. Our use of deep learning, this is our prediction. You see, our prediction basically have the same pattern as this one. And this one's other place, they didn't do experiments with this green color. but we actually can predict those places. We predict with some place they make things worse, some place make things better, things like that. So this is shows how mathematical tool, well, AI and the mathematical tool, how powerful it is. Yeah, we can basically replace the experiment with the same level of accuracy. With this technique, we can predict things. For example, this is a particular one is Eli Lilly antibody. This is the one is on the market. So there's a story about this antibody. It's an antibody therapy, okay? Eli Lilly by Eli Lilly company. So, and we look at the type of mutations, and those type of mutation, how this Eli Lilly antibody is going to be affected by the spike protein RBD mutations here, okay? All those mutations occur on this place. I just summarize here. And then how each mutation affects this antibody, I put it here. So it's look at a binding free energy change, right? So for this one, this is the binding free energy change in, so Eli Lilly induce like a weakened, just going this way is weakened antibody. That's by 2.7 times. But these ones, they are, for example, this one is the mutation, okay? There's a mutation, spike protein mutation at 449. It's going to reduce the Eli Lilly antibody efficacy by 21 times. And you see other ones reduce other different place, but this one is more special. This one is a E484K, that's in the beta and the gamma variant. So reduce the Eli Lilly efficacy by 55 times. That's basically make this Eli Lilly the antibody drug useless. And you can see this is what happened. So in the March, so FDA, they actually stop distribution of Eli Lilly coagulating antibody treatment due to the beta and the gamma variant, right? But just in the last month, September, so FDA again allowed Eli Lilly to go back to the market. Why? And you can see the reason, right? Originally was this guy, this one, they reduced the efficacy something like 60 times. But however, the Delta variant is dominant in the past a few months. And the Delta variant doesn't have 484K, right? It's a 484, it's a lot of 484K that goes to K. So actually it's a I think this is type 478, okay? T478K, this is a typo here. And also area 452R for those two, right? Those two are really for the Delta variant, yeah. So that means this Eli Lilly drug is useful for Delta variant. That's why U.S. government allow it goes back to the market again. Yeah. So this is a summary. You see all different variants in this slide. So basically, you see this is the data. So data has a biggest red color. So the red of the color means the higher frequency. So data variants dominate in all. The frequency is extremely high. And then you see the beta and the gamma, the color is around here. So gamma still has a lot of population. And beta has a good population there. And also you see lambda, the population is around here. So this is a lateral log scale, okay? So that color means that delta is the reddest one. And also there's the number, right? And also the height. What does this height mean? Height means binding free energy change. The higher here, that means the higher infectivity, right? So delta infectivity is high. Everybody talk about that. But it's a lot, it's much higher than the beta and the gamma, right? And the other one, there's a lambda mutation. The height is similar to delta. That means the infectivity is similar to delta. And there's one more thing to pay attention. This is the number 40. What is 40 means? So that means 40 is delta has the ability to disrupt the 40 antibody. Remember, we studied 130 antibody. data is able to make things like antibody escape mutations. So even you have a vaccine, and the data is going to destroy mostly like 40 out of 130 possible antibodies we studied. So that means the data has ability to escape a lot of vaccines, right? So, and also you look at this lambda, it has a 58, the number over there. That means it has higher ability to escape vaccines, right? You can see beta has 90. That means beta has a very, very high ability to escape. Unfortunately, we don't have very many beta in the, I don't know, at least in the US, but in other places, the beta is still there, right? So this one to pay attention, right? This is something, you see, everything is going up, but this one, the binding free energy change going down. That means they are making virus less infectious, right? So this is the one, you see. But however, the number is higher, the highest number is 94. So that means it's a vaccine. So what it means, okay? I put since here data pass, that means they are horrible infectivity, right? They are much more higher, the higher infectivity compared to data. And this one, I labeled it as a vaccine resistance mutation because they are transmitted among the patients, not through the infectivity, they through the ability to break through the antibody or vaccine. So that's why it's vaccine resistance mutation. So at this point, we have two mechanisms for natural selection. So one is the infectivity strengthening, right? The change in infectivity make it more infectious and they survive. And otherwise say, oh, if you guys have a vaccine, I have a new mutation such that it induces a mutation such that they have an antibody resistance, and then they can transmit among the people. So that's how it's the second mechanism. Okay, so I can actually show why this happened at this so-called vaccine resistant mutation. It's happened at a place, all those places I put over there, like Denmark and the UK. You see, those are the frequency, mostly in Denmark. And this is a frequency is with this red line over there in the Denmark. And this is correlated with what? It's correlated with the vaccination, fully vaccinated people, things like that, right? So vaccination. So Denmark is highly vaccinated. It's a European country. It's highly vaccinated. And so that's why this type of vaccine escape mutation become popular in Denmark and also in the UK. And most of all basically happen in the European country and the US. And those places, they have a lot of vaccination. And then those type of vaccine resistant mutation just take off over there, right? And this is come back to the picture I showed you before. Remember, I was enjoy something like all the top 100 top mutation, they are really infectivity strengthening up to the July of this year. And later on in August, we look at it again, all of a sudden give us a shock. And those two mutations, these ones are the vaccine resistance mutation, they come out in the August statistics you see there. So those, they come to the top mutations, become top mutation. That's the top 100 mutation I showed you before. So in the top 100 mutation, vaccine resistant, they're red color. That's they showing off. Okay, they just show up over there. So that's the second mechanism we discussed before, right? So I would summarize the things everybody knows at this point. The biology science is so important. We don't really fully understand the SARS-CoV-2. That's because our understanding of biology is still limited. So the last frontier of science is really biology. And however, in my opinion, last frontier of biology is mathematics. So if you're interested in the mathematics, doesn't matter what you do, like different topology, different geometry, different analysis, can do all kinds of different analysis, right? You can do different graph theory, and also statistics, probability, or differential equation, number theory, whatever. anything you do, it has a place in the biology. That's it. So mathematics is going to contribute to biology in a dramatic way from now on. So that's what I try to say. And this is basically my team. I don't want to spend too much time talk about my collaborators, my former students, postdocs, my group. So with that, I would like to give some time to take a question.
Am I audible, sir?
Yeah.
Yeah. So very nice talk, Professor V. So I have a couple of questions actually. We are also working at SSIHL. We have been also working on within host. So we are working more on the immune system, how it reacts with respect to the COVID, and see what are the different, what are the things, especially with respect to the drugs that needs to be given and optimal drug regimen we are trying to work on. So based on your work ahead, I just two couple of thoughts triggered. So we have you have identified multiple mutations, different variants at different positions in the genome. So is there a way based on it occurs at this position, delta variant at 254, whichever position, or the gamma, beta, different variants occurs at different positions. So one can be infectivity can be high, whereas the other variant infectivity may be low. So at the population level, based on this study, at the population level, can we say the infection can be extremely something like a second wave can happen because of this variant, because it is more infectious, or a third wave can happen because of the other mutation that has happened? That is the first question. And the second question that I have is, so how long these mutations go on? I understand virus is more intelligent than humans. So these mutations, does it go on infinitely? So where does this COVID cycle end? Or is it that the mutations are going to happen and we have to keep on continuously producing different vaccines to encounter these mutations? These are a couple of thoughts which triggered in my mind when the presentation was going on. Over to Professor V.
Yeah, very interesting question. Thank you. The first one is so we identify those variants over the this is somehow we look at variants, their infectivity and also their ability to disrupt our antibodies. Those are two things we studied. But related to what you are doing over there, you look at all kinds of other immune system, and they are hugely complicated protein-protein interaction systems over there, and how these mutations and other mutations I showed in earlier, like 28,000 mutations, affect those things over there. Our whole immune system we really don't know very much at this point. This is something we need to study, and we also nobody can study in the detail because too many mutations, 28,000 unique mutations. So it's basically almost like a mission impossible at this point. And there's definitely, if we know the science more and biology more in the future, probably we have a better ability to analyze the detailed each individual mutation, how to affect the other things, like a virus ability to induce like a cytokine reaction or something like that. At this point, we don't really know much, right? Things like that. On the second point, second question you asked over there. This is a really good question. That means we are going to keep it to have by two mechanisms I will show here, right? One mechanism is generate a lot of more infectious mutations, so they have ability to propagate. Other things, they are going to have a lot more like a vaccine resistance type of mutation to show up in the later. So that means this whole thing, in my opinion, okay, the whole thing is going to continue for many years, but the whole thing is going to, after all, the population, everybody get either infected or otherwise they get vaccinated, and this is going to become other flu virus, something like that. And we're going to every year we have flu shot every day, so every year this is going to be the situation in the future. we are not going to stop this virus totally. That's impossible. We lost such a point of control.
Okay. Okay. Thank you, sir. Sir, as I requested earlier in our emails, can you just tell, because being a maybe not this regular COVID discussion, but being a managing editor for the journals, so can you tell a little bit, because we have a lot of participants who are keen to publish in good journals. So what does the editorial board look like, just for the participants benefit? Maybe just 5-10, because we have time till we have a few 15 minutes more time, so maybe 5-10 minutes time you can spend, give a little insights on that. It will be really beneficial to all the people who are attending this conference.
Yes, thank you. I prepare one slide, I put it here. So I do a lot of editorial services, that's true, and I try to put an editorial process, just put it in this way. And there's the first thing is that when you submit a paper into a journal or something like that, there's a professional screening. So the people call it professional people, the paid. They are not don't have a knowledge of detailed what you do, but they just for the journal. So there are those people, they look at it. For example, they check also list if there's email address, everything's listed over there. They can contact you, things like that. They look at your submission material, right? They look at the manuscripts, page limit, figures, and the tables, supporting information. If everything looks like a file or something like that, they check those things. Particularly good paper, a good journal, they have a page limit. And also they have a number of figure limit, things like that, and the number table limit. For example, like a journal Nature, and also the Nature company, other journals in the Nature company, something like that. They all do that. They tell you to make change. So the action is they return for resubmission. They say, okay, you have too many pages, well, you too many figures. They ask you to, they just send back to you, things like that. So, and then mostly at this point, good journal, the journal about allergy or something like that, the good journals, they have a data set and code availability. They want your data set to be posted somewhere, and the code availability, your code is deposited somewhere. So this is for the reproducibility. So more and more journals, they emphasize this. In the old days, we just tried to publish some things over there, and nobody check our data set. Nobody check our code if they are working or not. But this is no longer the case, particularly for some good journals, right? So, and also they check similarity, right? And also dual submission. So if they see you have a dual submission and your paper turn out in other different web page or different journal, they will just basically reject your paper. I mean, those editor office people, they don't make such a decision, but they take a load. They put a warning over there. If you are similarity, even the similarities with your own paper, that's not good. They're not allowed. If it's higher than 30%, they will put a warning over there. So this is as a journal office, typically the secretaries over there, well, the professional people over there, what do they do? And then there's after that, they will, if everything is fine, they just give the manuscript to the editors. And typically, some big journals, they have an editor-in-chief. Small journals, they just have editors. So those editors, they will look at it, and also based on the suggestions from a journal editorial office, they are going to they are looking at the suitability of the journal for the journal publication. First, they are reading a title and abstract, and to see if we are falling to the scope. So they see, okay, if this is my journal is particularly for something, I want to see this type of submission is somehow aligned with my journal mission or something like that. So that's things they're looking for. They're also looking for style and quality, right? Quality means what? Because I mean, you have nowadays, there's a science and the nature. Those journals, they are different. Why they are different? Even those journal, the editors over there, they are going to change your paper just order by order. They are going to write it. If they accept your paper, they will write the paper for you. They basically write a paper for you, okay? So, but for other journal, I mean, just ordinary journal, majority journal, the editors, they are very busy. They handle like a few hundreds of papers per year, something like that. So in that case, they don't really have time to read your paper in detail. They look at the title and look at the abstract, and they look at the figure and the table. Those are the four things they look easily, spend like 5 to 10 minutes, and to see if there are something to their taste. If there's something to their taste, and they look at particularly figure quality, they immediately see if your paper is good quality or not, and then they make a decision, right? And the potential popularity, sometimes they even look at check the author's citation history, things like that, and also the topics. They wanted to see the topics is going to be potentially have a lot of high citations, things like that. They do pay attention to that. And then similarity, remember the journal office already checked for that. They see if similarity is too high, particularly with the earlier publication, they will reject. And the reference limiter, safe citation, they don't want a paper like, say, you have a 40 citation and 20 of those citations are just your own paper. They don't like that. So they also don't want you have an ordinary paper like 100 citations or that's too many. So they make a decision. That's just an average journal, right? So for example, an average journal, they will immediately reject 30 to 40%. And then sometimes they think, oh, something they feel unhappy, want the author to fix it. So that's 2 to 5%. And then they send the rest to the reviewer, about 60%. This is just an average reputation journal. So editorial decision, okay? So for example, this is a journal, I'm an associate editor. This is impact factor is 5. It's a journal of chemical information and modeling. And the other journal, I'm an editor-in-chief, IGNMBE. This is the International Journal of Numerical Mass Biomedical Engineering. Impact factor is 2.7. And based on those two journals, so basically, we at least 2 or 3 reviews, mostly 3 reviews. Sometimes we cannot get 3. We also settle for 2 reviews opinion. Basically, we rejected something like 35%. Remember, here I already rejected 30 to 40%. Here I rejected 35%. And then rest I can put a different option, but all this rest option is going to eventually publish mostly, okay? So reject and resubmit. If you take care of your reviews, comment and suggestions carefully, you typically get accepted, right? So this is the 30%. Major revision as also other 30%, you see? So, and then we don't really, minor revision 5%. And we don't really accept anybody's paper at the first review process. So that means you get rejected, there's no more chance. Anything you go to here, there's a more chance for your paper to come back. So that's basically editorial process. Any questions?
Thank you. I don't think so. There are any questions, sir. So since there are no questions, there was one comment from a participant in similar lines to the question which I posed. So the question says how to kind of choose the exact population model or a population model based on within host. Maybe because we're running short of time, I would like to state that there is a different kind of modeling which has taken over in maybe last 40 years. It's called multi-scale modeling, where you see the influence of within host, how it actually affects at the population level, because the time scales are different. Within host, that way in which the virus multiplicates or replicates, it's much faster compared to the way in which human multiplicates or replicates. So you have to get both of them in a single time scale and work on a reduced model which involves both within host and vector to host. So that we have actually just started working on. Tomorrow you can listen to us. So we will so since there is not much of time, So let us all thank Professor V, not only for the wonderful talk, sir, and it was like really enlightening to see how mathematics can be used to understand the different, especially the algebraic topology, what you have seen, the branch of algebra, how it can be used to understand the different variants for this SARS-CoV-2. Thank you, sir, for
Thank
your
you.
talk, and also for the editorial insights into the editorial process that undergoes as soon as a manuscript is submitted. Thank you, sir.
Yeah. Thank you. Thank you very much.
Thank you, sir.
Yeah.
So to introduce the next speaker, I request Dr. Khanna to come over to the dais.
Good evening. For the next keynote address, we have Dr. Edwin Michael on forecasting the future paths of COVID-19 pandemic using adaptive data-driven simulation. Dr. Edwin Michael is an epidemiologist who studies the spread and control of global infectious diseases. The overriding objective of Dr. Michael's research is to address the next generation of critical questions regarding epidemiology, population biology, and computational disease dynamics. Dr. Michael did his MSc in zoology from Madras University in 1984. He went on to do his PhD in parasite epidemiology from Imperial College of Science, Technology and Medicine, London, 1990. He was a member of British Society of Parasitology and British Society of Immunology. Dr. Michael was a professor at the Department of Biological Sciences at the University of Notre Dame from 2011 up to 2020, after which he moved to University of South Florida, Tampa, as the professor at the College of Public Health. Currently, he leads the development of Scir COVID-19 modeling portal for supporting population-level response and hospital surge planning to contain the pandemic at the county level. The Scir COVID-19 forecasting and planning portal provides unique, locally applicable epidemic forecasts. Without much delay, I now invite Dr. Michael to address the gathering. Dr. Michael, over to you.
Hello, hi, good evening, right, in India. Okay, so I'm going to share my screen. Share screen. Okay, can you see the screen?
Yes, sir.
Okay,
It's visible.
right. Okay, so thank you everybody for inviting me to the conference. It sounds very interesting, and I think as you rightly pointed out, and the organizers, we need to learn a lot about the modeling enterprise, and COVID has given us a fantastic chance, to try and learn how best can we build models. And sometimes you have to build models very rapidly and with very little data to parameterize the model, calibrate the models going forward, right? So this is our concern when we first started building Scircast, for example. We thought we do have the compartmental models. Should we add more complexity to the models? And then we considered that we have to work with the publicly available data, which might force us to rethink about the structures of the model, because we have to take the data to parameterize the model. So the data in some ways is driving the kind of structures that we need to think. And also at the back of our mind is heterogeneity in the system. How do we best capture that? And then down the line, we also thought we need to build an agile system which is adaptive. The structures need to be agile because we have to take into account that things are going to change in the future. And this is what I'm trying to talk about. But here my forecast will be what will be the future paths, of the COVID pandemic. given the kinds of models, modeling platforms that we have built, which are data-driven, taking data to inform the parameters, and then use those updated models to make forecasts. So the premise was mathematical models. When we first started, virus transmission dynamics, as we all know, can play an important role, for response planning and combating, managing, the current COVID-19. So we started building this model in March 2020. And we thought SEIR models should be the first basic types of compartmental models that we need to think. And when we put these models together, we thought that it needs to account for local transmission conditions, locally applied interventions, and then only those kinds of models can give us reliable forecast of the course of the pandemic. We also considered that these models need to capture rapidly changing social behaviors, ecological factors like arrival of variants, and then changing intervention policies through time. And that also needs to be captured. So then we considered, how do we put together a modeling system that can do all of this? And so we started looking at data science approaches to bring in data which are driving these models, and then a well-defined model which is agile and adaptive to the kinds of data that we can get, which are publicly available. And also we thought over the future, and what would be the anticipated changes in the data inputs as well. So how do we build a very flexible, agile modeling system, and to build a practical tool for tracking and predicting the future course of the pandemic? And we use sequential Bayesian calibration methods that allows updating a models as data on the outbreak evolves, together with estimation of uncertainties that need to be run in order to track and predict the future course of the pandemic. And this is the kinds of structure that we thought. We all are aware of this kind of compartmental models, so I don't want to go into the details, just to say that you have susceptibles, they get exposed by being in contact with infectious people, and then the infection can take two pathways, which is unique to COVID. One is infectious asymptomatic, or more unique to COVID, I would say. And then they lose infection and become recovered. And then you have the clinical pathway, infectious pre-symptomatic, mild, hospital, ICU. I mean, this is well, everybody knows this, right? And then we have, because of the lockdowns, we need to capture restricted movement, right? The rate of which people are put into restricted movement and then they are released. And then we also have this phenomenon of vaccine refusing people as well, okay? And the big circles I drew, these are where we have data, some of the confirmed infectious, confirmed cases, and then deaths. That is where the model needs to be fit, and then all the other parameters are going to be estimated. But we have two other parameters, which is D. Here, this is the impact of social measures captured by one particular parameter. How do we get data to inform this, right? And then the movement data. And then we consider that the structure needs to be modular by design, because things are changing all the time. We might have to add more and more compartments. The first thing we added was the two-stage vaccination, so two more compartments. And then the next thing that we added was explicit modeling of the alpha delta variant, which began to emerge. So, the alpha delta variants. And then, you have breakthrough infections coming from the vaccine classes as well. And then we have, lately the waning of immunity after you get the second booster, and then the advent of the third booster as well. So if you look at it, we, in our mind with it, we thought, look, we have only few data, infectious cases, deaths, and then some of the driving data. But we need to build a model in a modular kind of fashion by which it allows us to add these complexities as we go forward easily. These are the data sources that we use. Luckily, one feature of COVID has been this explosion of data repositories. That has made modeling this much easier, right? The data is not highly heterogeneous. For example, you cannot capture all the heterogeneity at the local level, but at the population levels, it gives us a lot of information. at the county level sometimes. Some data gives you at the country level. Some data give you also at the state level. For example, coronavirus app, gives confirmed case, death, and vaccination. So one of the enterprise, one of the things that we did was bring in data scientists to go in and pull this data. And this is how we model mobility, people's movement. The amount of people in lockdown or restricted movement, right, is captured by these two parameters, which puts susceptible people into restricted movement and then releases them. And we are informed by using this unicast data, which is tracking mobile phone, GPS data, and they've classified it into different types of, movement. Here, for example, we use change in non-essential visits to try and to get parameter values for this. So we fit to this kind of data to parameterize the movement of people into and out of lockdown policies. And then we use Google Trends data, search terms like, COVID mask, and this is this red line, and this is the average, a fraction of, of people who are interested in those kinds of search terms. We use that as priors to inform the, the estimation of D, the amount of social measures which are applicable in a particular setting. And this is the vaccination, the CDC vaccine tracker data gives us all the information at the county level in the US, but also we got, our world in data, websites that also gives us vaccination data at the country level all across the world. And this gives us, the advent of the variants. This is the Delta one. This is all other variants, for example. So we know that we can get all this data together. And this is the kind of coding blocks that we built. The first one is to pull in data, and we'll see later on this is automated. This I'm giving an example of pulling in data from Johns Hopkins database for all the counties in the US. We can pull in, deaths data and then confirmed case data, number, and the population size, and the timeline, that the data was pulled in. We push all that data into the mathematical model, and this is a snapshot. And then, this model is much more extensive right now. And then this is the calibration area, and we use sequential calibrations. So the parameters that we estimate for a previous time frame will act, those posteriors will act as priors for the next fitting time period. We start with 500, it's a Monte Carlo kind of simulation. We start with, 50,000 parameter sets, and then we use the sequential RMSE to select 500 parameters for each time window, right? And those are our ensemble that we use to make forecasts into the future. And then we have all the output coding. And then we started building this continuous integration, and this is an automated system to try and automate the pipeline. So the data is coming from also, we have API, and custom scripts to go and pull all this data. The continuous integration will check if the data is new, it'll start, we have a routine to clear and prepare the data as for inputting into the models. The data is pushed into the models, and then we do the calibration, the sequential calibration here. So we have priors from the previous fit, which will serve, the posteriors from previous fits will serve as priors. And then the fitting, the calibration method will give us the posteriors, and then we use that to select, right, the ensemble model, and then we make the forecast. Okay? And all that is automated. And this is simply showing the important sequential fitting to incoming data. Let's say we have a data stream here, the open circles and the open circles are data. And the closed circles are, let's say we fit it to the first 40% of the data. And then we make projections. You can see we're not able to capture the future. If you fit to the next tranche of 20% data, so the first 60%, you're beginning to capture the future, right? But there's a lot of variance. 80%, you shrink the variance, and then so on and so forth. So the importance of sequential fitting, because things are changing all the time. And this is the beginning of our forecasting, right? I mean, we've done this forecasting all across the US, even for India, for Cayman Islands, I'll show some results, etc., because it's so easy to, once we have this data-driven system, as soon as we get the data, we can fit the model to the data and then make projections. This is the for Florida, right? And you can see the pandemic has gone through several cycles, and this is typical in response to social interventions and then vaccinations, etc., etc. You can see that as soon as you bring in an intervention, that will have an impact. For example, first lockdown, you suppress the wave. As soon as you release the lockdown, the wave, we got a big resurgence, last June. And the data is the solid line, and the model predictions are given by the dotted lines. So the model and the data are responding to social measures, and this gives this characteristic cyclical, transmission that we've been seeing with this pandemic. And since December onwards, initially we had the original variant, then the alpha variant began to play a role, the blue line. The solid one is the actual cases contributed by the alpha variant, and this is the model predictions of our, the variant-specific model predictions. And then the first observation of the Delta variant began in the end of April, somewhere here, and then you can see the model is beginning to capture Delta variant as well. Sorry, Delta variant started in April, the yellow line, and this is the original, and I think that was the alpha variant, which is here, right? And then we'll see that this is the blue is the original variant, the Delta and the alpha variant, and this is the Delta variant. And then and you can see the data and the model is matching the data, very closely, and that is because of this sequential fitting that we do. And then we can make the the near-term forecast. For example, for Florida, the short-term forecast is really good. Cases are coming down. So how do you make the future path of this pandemic? What kind of paths can this pandemic take? For that, we need to keep an eye on herd immunity, right? And one of the things about this kind of modeling is that we can get at the latent variables, and one of which is the amount of people who are developing immunity to the virus. That is the black line, and this is a susceptible fraction. And you can see right now, the Florida population, about 88% are currently immune. And then herd immunity, we are now estimating is going to lie between 90 and 92. So we are very close to herd immunity, right? And it's only about 9% are currently susceptible. And this is interesting. We can also take this model and work out, what fraction of these immune people, what fraction comes from vaccine-conferred immunity and what comes from natural immunity. This is true infection. So right now in Florida, 54% of all immunity comes from vaccines, and natural immunity is contributing 34% of the total immunity, the 88% that we have achieved. And we'll see the importance of this later on. So let's look at the first long-term, path of the pandemic. And, the pandemic, the last wave peaked August 22nd, 21,772 cases and daily cases, and then it started dropping. And this is the future. The solid line is current social measures and vaccination. If that were to continue, and if immunity was long-term or permanent, the pandemic is going to have a long tail, declining tail, and eventually will fade out. This is really good news. And then we also looked at simulations looking at full release of social measures from now onwards. That's the blue curve, nothing much. And then what if we increase vaccination? School vaccinations are going to begin. What impact will that do? Very little. With full release or with the current social measures, very little difference. The reason being, we are very close to herd immunity. So very few susceptible people are there. So these extra interventions are not going to make a big impact, right, going forward. And the interesting thing is, so far, this is the period we are in, very little sign of resurgence, indicating that, the social protection and the current vaccination are holding the pandemic, in place, so to speak. We can also take the ensemble models and work out the fade out probability over time, right? And the blue one is with the current social measures, okay? And the way we do this is we use moving windows of like 21 days every time, and then we look at the rate of decline. the fraction of curves, the 500 curves that are showing decline in cases over that window. And from there, we can work out the fade out probabilities. The blue one is showing that fade out probability. It will be reached if immunity is permanent, and July 12th, most of the curves will be showing, declining slopes. But we can also show what if we can easily manage 1,200. That is, how many of the curves, can, go below 1,200. If you use that threshold, we'll reach fade out January, early next year, 2020. This is the corresponding thing. If you fully release social measures from now onwards, that will push the fade out to later next year, the blue curve. And then the red curve is showing in the middle of next year if we use the 1,200 threshold. So what other things can happen in the future? One is the waning of natural and vaccine-conferred immunity. And here we're showing simulations. What if immunity wanes over 1 year, very fast waning of immunity, 2.5 and 5 years, those curves over here, and then no waning. The first thing you'll notice is there's waning of immunity immediately. You will not get this long-term, long tail, fade out tail. Instead, you will get cyclical pandemic resurgence. The size of the coming waves and then the interwave period will depend on the duration of immunity. If immunity lasts longer, if waning occurs over a long period of time, 5 years, the size of the wave will drop, and also interwave period will lengthen, and that is, gives you a window to intervene. But what is the sign that immunity might be waning? What we did is we fitted, all these models to data, around this area, looked at the RMSE, the fits, and we show the better fits are given with models with waning. And it reduces the prediction error, up to 12.4%, with waning immunity. That is indicating that we are getting the first signals of waning immunity. But the best fitting model is the 5-year immunity, waning immunity model. So immunity might be waning, but it is quite, it's going to wane over a moderate long-term period. The reason for that is, we'll never reach herd immunity, if there's waning. 1 year, this is the immunity level, 90 to 92%, we'll never quite reach. And that's why we're getting this resurgence that we're seeing for 1 year. And then for 2.5, once again, you're approaching herd immunity, but it'll fluctuate. Similarly, for 5 years, you might get into the herd immunity regime, but then it'll start fluctuating. And that's why we're getting these cyclical waves in the future. And then our future paths, what can we do if there's waning immunity? Yes, we can look at booster vaccination. As soon as you have booster vaccination, which is given by the dashed curves, this is for 1 year waning, you can reduce the size of the coming wave by giving the third booster, right? But if you give the third booster also for 1 to 2.5 and 5, you can fully suppress the coming wave. So the boosters might be a way of coming out of these problems of waning immunity. Here we are bringing in the advent of more transmissible variants. We're looking at 50% more transmissible, 10%. This is the AY 4.2, which is spreading in the UK, for example. It's thought to be 10% more transmissible than Delta. And this is a base case, the black line, right? And look at what happens. If it's 50%, you might get a significant, wave occurring, which will occur, in the middle of next year. And then 25%, you'll also get a wave. But 10% is hardly going to do much difference, in terms of the coming wave. But if you bring in booster, you can collapse. You can suppress all of these impacts. And this is what if it's transmission, a more transmissible variant comes in with 1 year waning included. And this is 1 year, you'll get a big spike. But if you bring in a booster, you can reduce the spike. But if immunity were to wane for 5 years and you have the booster, you can suppress this. Okay, and so that is the pathways that we can expect, going forward. I'll summarize in a minute. We also looked at Cayman Islands, their post-vaccination wave of cases. which are occurring in Cayman Islands, in Vermont, in some other states in the US with very high vaccination rate. And this is despite having reached very high levels of population immunity. In Cayman Islands, herd immunity is about 84.5%. Currently, they are 82.8%. But the difference is this: for Cayman Islands, vaccine-induced or conferred immunity is very high. 79.6% of all this immunity, current immunity, is coming from vaccine-induced immunity. A fantastic vaccination program. But they also had very strict lockdowns, so very little natural immunity. For Florida, 54% is vaccine-induced immunity and 34% for natural immunity. And this could play a role, because there are some studies showing a natural immunity might be stronger than vaccine-induced immunity. And that could be the reason why we are seeing a flare-up of cases in those states. The other thing is, breakthrough infections also pretty high in Cayman Islands because there's a lot of vaccinations. So in summary, data-driven compartmental models, sequentially updated with publicly available data, can offer a practical tool for tracking and predicting the course of the pandemic. We predict several future paths are open to the pandemic over the medium-term future, over the 1 year ahead kind of future. If immunity is long-term and permanent and no new variants emerge, then one future is for the pandemic to burn out by mid to late next year, depending on levels of social measures and vaccination. This is great news. But if immunity wanes and more transmissible variants arrive and spread, then cyclical resurgences are possible, with size of the resurgent outbreaks and inter-outbreak period dependent on the duration over which immunity wanes and the transmissibility rate of any new variant. We also indicate that for these two futures, administering the booster vaccines, which may need to be repeated, will allow control of the pandemic and hence safe return to normalcy. That's it.
Thank you, Professor Edwin Michael, for the wonderful presentation. So I have a quick question listening to the presentation. So in the present day, in India, we have a Covaxin or COVID Shield, or in abroad, we have a Sputnika or Pfizer or AstraZeneca. Different vaccines are available. So the efficacy of the vaccine is different. So in your model, when you say you give the vaccine, it has the waning immunity levels. You're doing that.
Yes.
So when you say vaccine, are you really considering even the efficacy? Because the vaccine is kind of a general term, and we have multiple vaccines.
Yes.
So if so, what are we actually referring? Is it a generic vaccine or different with respect to different?
No. You're absolutely correct. It's hidden in the. I didn't show you the details. Yes, we consider the efficacy of the different vaccines, and for Florida, we're looking at Pfizer and Moderna, in particular. Okay. So we bring in those effectiveness of those vaccines explicitly.
And also the coverage is like
yes.
The coverage aspect also you have been taking.
Absolutely. Because we cut the vaccination rate. Yes.
Yes. Actually, we at SSH also we are working on the different vaccines and the coverage strategies, not from the data-driven approach, the traditional deterministic approach.
Oh, I see. So we take the data. We take the data to inform the coverage. Yes. And then we bring in the effectiveness of the vaccines as well.
So we are. So what we have been doing is like for this kind of coverage, this is how the death rate or the infection rate. So we are trying to predict, and in fact, we are trying to validate as like a future trend.
I mean, for your information, Anuj was also part of this. We did look at, when the the May outbreak in India, right? We fitted the models to India. And we also look like what you were doing. What will happen if you go immediately into lockdown? What will happen if you increase the vaccination rate? So there are two things that we can do, right, with models. One is we can ask this kind of scenarios.
Yes.
Like you were saying. The other thing is actually take data to inform the model and make it, much more realistic, inverted commas, to the local conditions, transmission conditions.
Yes. Retrospective and prospective.
Yeah.
Yes, sir. Thank you. Dr. Anuj,
well, hello, Dr. Michael. How are you doing? So just want to tell the participants and audiences that please do go and check out the sercast.org website, where Dr. Michael's this dashboard is there, which was real-time information that and his results were actually used by some of the Florida departments, and they were basing their policy based on that. Additionally, I just want to add few more things. He has worked for many years on lymphatic filariasis, and filariasis, I think, right? What in India also, in India.
Yeah, absolutely. Yeah.
And so if you have any ideas, projects, something that you want to discuss about, it's Professor Michael is.
And we also, under you were part of it, and we also were looking at urban dengue, in India, once again taking a data-driven approach, as well. Because the problem is, we can build this model, but you have to constrain it. The parameters need to be constrained to capture local, conditions. And that has been our principle driving these models. Otherwise, it will be theoretical exercises. And that's why policymakers will lose, faith in these models, because theory is theory, right? We are asking, what might happen given the parameters that are driving the model. And sometimes, remember, initially when the initial predictions were made by Imperial College, right, it was ridiculed at the end because none of those things played out. And this is the problem with theoretical models, because they do not capture transmission conditions on the ground. And so we might lose the trust of policymakers if we don't confront models with data. So the data will constrain the model. Yeah, correct.
Can you, Dr. Michael, can you talk a little bit about, touch base on, when you say connecting with data, there are many people who are doing connecting with data, but your way of connecting is very different, because you are doing something like nowcasting. Can you kind of throw some light on that?
Yeah, so, the data itself, cannot be taken as given, right? There's a lot of reporting errors. There are other kinds of lags in the data. So we bring in a nowcasting approach, and if you look at now, that will adjust for these parameters, this kind of heterogeneity in the data, so that we can get, so we are adding some, some mathematics into this, but that data is much better because it takes into explicit account into data reporting delays, gaps in the data. If you look at many of the repositories, people dump data after 2-3 days. Like in Florida, you get 0, and then a huge dump. So the nowcasting approaches can correct for all of those as well. And we use that to fit the model.
Yeah. Yes. So there's a lot of...
Yes.
Go ahead,
Yes. There's a one quick question from the participant. So when you give these boosters, booster vaccine, so is your models, do they incorporate the side effects that can happen because of these boosters? Because we don't have data.
No, we don't have data. Yeah, exactly. So I've just got some information, working with some people from Jamaica who exactly asked this question. So we're beginning to start looking at the data, but Jamaica, 751,000 vaccinations have been rolled out, and the adverse effects is like 497 cases, tiny, cases. And I suspect that will be the same in many countries, like India, for example. I don't know, what is the vaccination rate in India? I think it's exploded now, right, in India?
Yes, sir. In the last few days, almost we have reached to more than 70%.
Yeah. And so but you're absolutely correct. Nobody's tracking that well adverse events.
Yes, sir. So just a couple of days back, India, we have almost reached the milestone of 100 crore vaccinations. Sir.
Yes. So India's exploded since May. When that big wave occurred. Yes. So the Indian government did the right thing, pushed vaccination quickly, and that was our simulations as well. Anuj, you will remember that we have to ramp up vaccination. That's the fastest way to control this pandemic.
Yes.
But you're right. Adverse events is still up in the air. I mean, the Jamaica data is the first one I've seen where they've really collated. But it's a small island, right? For India, how we can get the data across all the states, that's a challenge.
Yes, sir. Yes, sir. Yeah. So, if Dr. Anuj, do you have any comments further?
I think with the show, we don't have much time, so we want to thank Dr. Michael.
So, Thank you.
Thank you,
Professor.
everybody. And this is a really good conference. I'll wait to see your final conclusions. Yeah. Thanks.
Yeah, and we are on the YouTube also,
Oh,
Dr.
good.
Mike.
Yeah. But I think India, you got the mathematicians, you got the physicists, you got the core group of people. But what is missing is disease modeling as a major, kind of. I mean, it's not taught in universities, as far as I know.
Yeah.
So I think this kind of conferences are really useful to change universities thinking. How you can bring in, like my postdocs are all from physics. They are coming from mathematics, pure maths, but they are interested in biology because biology is so fascinating, right? So they find it even more interesting than doing physics. So they come into biology. So how can we build this kind of system in India? Is it's a question that we need to address.
Yes, sir. That is precisely we have taken the first step. So we have a lot of steps to go and
Yeah.
climb the ladder. So we hope to take it further, sir.
Yeah.
Not just at this conference level, but we want to take the collaboration much further, sir.
Yeah.
Dr. Michael, just one last thing. Do you still have the opening for the postdoc position that?
Yeah, it is open. It is open. So please, Anuj, if we can, I send you the write up, right? The advert. If that can
I will
be
share
passed to the conference people. Yeah,
Sure.
absolutely. But I think last thing, but in terms of Indo-US, there's a lot of interest. Even from the state side, right? Or working with Indian scientists. Like with Anuj, we wrote this proposal for dengue with people from Pilani, right? Birla Institute. And those are physicists, and also from Delhi University. So those kinds of, we need to push the government to also have those kinds of programs that can encourage India-US collaborations, and also globally, right? Because more tradition in mathematical modeling of diseases occur in the West for whatever reason, So I think, but India's got the brains, and the people, and the data is a gold mine, in India. If we can bring all this together, I think you can revolutionize, disease modeling. With that, I shall quietly withdraw.
Yes, but it was really encouraging, sir. It inspires us to really take this further. So as we are really running short of time, I request Dr. Khanna to introduce the next speaker. Thank you,
Thank you,
Dr.
everybody. Thanks.
Thank you, Doctor.
Thank you, Dr. Michael, for the wonderful talk. For the next keynote address, we have Dr. Lance Waller on using what you have, infectious disease surveillance with limited data and time. Dr. Lance A. Waller is professor in the Department of Biostatistics and Bioinformatics, Rollins School of Public Health, Emory University. Dr. Waller received his bachelor's in mathematics from New Mexico State University in 1986 and his PhD in operations research from Cornell University in 1991. Professor Waller leads the Woodruff Health Science Center's strategic initiative in data science. He is currently a member of the US National Academy of Science Board on Mathematical Science and Analytics and serves as co-chair of the Committee on Applied and Theoretical Statistics. Professor Waller was recently appointed to a 3-year term on the US Census Bureau's Scientific Advisory Committee. Dr. Waller's research involves the development and application of statistical methods for spatially referenced data, including applications in environmental justice, neurology, epidemiology, disease surveillance, conservation biology, and disease ecology. Dr. Waller has published in a variety of biostatistical, statistical, environmental health-related, and ecology journals, and is a co-author with Carol Gotway of the text, Applied Spatial Statistics for Public Health Data in 2004. Without any delay, now I invite Dr. Waller to deliver his keynote address. Over to you, Dr. Waller.
Thank you very much. I will share my screen here. I'll have to move the file over to the screen and we should be set in. So thank you very much for the kind invitation to participate.
Dr. Waller, can you share your screen, please?
I'm trying to. It says the browser can't access your screen. Let me try again. I'm not sure why it won't allow me to share the screen.
Dr. Waller, there's a share button. You need to click the share. So it says then you have an option of screen share. You have to press that share.
I can get that far, but when I get to it, now it asks me to select a screen. I click share screen, then it says select window or screen.
No, entire screen. When you see there are three options, you need to select the entire screen option, the first option. A window, you see the three options. You select the first option, entire screen.
Then all three of them say browser can't access your screen. Let me see.
Using what browser you are using, Dr. Waller?
Using Firefox because it wouldn't open on Safari.
You have Chrome? I'm using Chrome actually.
Yeah,
Chrome.
probably have Chrome on here.
Maybe from Chrome it may be a little better.
Let me try. There it is. My apologies. Let me switch over to that.
No, not at all, not at all.
Good thing my title is using what you can.
Somehow do you have to close this browser, Firefox?
This is Enoko. We will wait for Waller to join.
Yep. We can see you back.
Oops, sorry, I'm on the wrong browser. Wow, that's a good first impression, is it not? All right. Let's try it through Chrome. Share screen. There we go. That looks more promising. Pull up my slides.
Entire screen, and you select the tab and say share. That should be fine.
Okay, can you see those now?
Yes, perfect.
All right, I'll put it.
Okay, you can make it full screen. Yeah.
Yep. Okay. Again, thanks for your patience, and thank you for the kind invitation. So the previous talk was really fascinating, and the intersection between mathematical modeling, epidemiology at a health department, and data collection has really been brought into stark contrast during the pandemic. A lot of us worked in our own silos related to infectious disease, but trying to answer questions quickly, accurately, and with impact has proven very challenging. You don't have a lot of time to give an answer, and you need to use the data you have. And I want to think about some of those data things and communicating complicated ideas to the general public. So I want to say a bit about public health surveillance, pattern and process, alternative data sources, and then think a little bit about testing in the background. So public health surveillance is an old idea. It's been around for a long time, and this is sort of the standard definition: the ongoing systematic collection, analysis, interpretation of health-related data essential to planning, implementation, evaluation of public health practice. So this comes from a mindset of you can only get the data you can collect. And now we live in a data science world where we grab all the data we can get and we try to see how this helps. So the Centers for Disease Control and Prevention are next door to Emory, so we work closely with them on there, and they've had an ongoing, long-standing history of public health surveillance. You'll see this slide over and over again. My students get very tired of it, but this is how this is really taking the scientific process and then just bending the linear process into a circle. We start up on the upper left with questions we want to answer. So in a surveillance setting, where is the disease? Where are there active hotspots? Where is vaccination coverage high? And you see, I think of all this stuff in space on maps. Then before we do anything, we can move down to what data would we need to answer the question. So one of the maps people wanted to see throughout the pandemic is what is the local prevalence? But we don't really collect data that tells us that. So we get reports on percent testing positive, and nobody's kind of happy with that answer, but that's what's available. So the data we would need for prevalence would be something like a random sample or a sense of how many people are infected over how many people are at risk. But the data we can get is often different than that. It's how many people came in to get tested. And if you have university students getting tested frequently, like we did last year, they're going to be overrepresented in here. And if you have individuals in the public who are maybe hesitant to get vaccinated or anxious to get out and interact with others, but they don't want to get tested, they're not going to be represented either. But now once you have the data you can get, there are questions you can answer, and that's where a lot of reports on percent testing positive were showing up, but that really wasn't the question we started with. It was close to it, but what were the relationships between the question you do answer and the questions you don't answer? And then that also can help you. How close did I get? What I need to get closer? What data can I get? So you can work a little harder and you can make your way around and around this process. So it's not really whether you got the answer, the final answer, but whether you know more now than when you finish than when you started. So public health surveillance can be a tricky goal. It's different than a scientific study in the sense that there's multiple goals, there are multiple constraints, multiple data types, and multiple users of the results. So let's think about these somewhat. The multiple goals, like the overall goal of any type of surveillance, is situational awareness. What's going on now? What has been going on in the past? What may go on in the future? We might be concerned with incidence. Where are the new cases happening? We might be interested in prevalence. Where are the existing cases? Because that'll fuel the risk in an infectious disease for the new cases. We might know where are the infections. And so case finding is, we're all familiar with, but takes a lot of work. Contact tracing. Where is the map of what is the map of local vaccination coverage? You think of a fire burning across a paper map, maybe there's some spots with gasoline on it where it would go much faster. When are the infections? When somebody, one of the insidious things about COVID-19 is you are pre-symptomatic and infectious. So you don't feel sick. And so self-regulation of interaction, social distancing, and so on is not very effective because you've already done the damage before you feel bad. On the good side, on the prevention side, the people who are going to be infected 2 weeks from now are not yet infected. So changes you make today can make a difference. What resources are needed where? You'll remember early in the pandemic in 2020, getting healthcare workers. Now we have healthcare workers who are very tired of dealing with the pandemic and burned out. So where are people? Can you move them around? Can you move professionals to where the biggest problems are? Where's the personal protective equipment? This was at a premium early on. Which beds are available? Every surge we have where the intensive care units become full, there's a resource question that's a big problem. What's going to happen next week? What's happening next month? What's happening 6 months from now? How far out do you want to project? And then what's the effectiveness of the response? How well is vaccination working? What's your local coverage? How is that impacting the number of cases and so on? And how are things going to change when we have treatment options to roll out? So we're seeing some new approvals of drug pharmaceutical treatments. There's a lot of constraints on running a surveillance system, money and time. It costs money to collect the data, and how fast do you want it, and how accurate do you want it a day later? So more money would give you shorter time. And then what's the role of testing, and we should think about what the role of long-term testing for, let's say the rates do go down in the spring as projected. Do we want to keep monitoring? How do we find it? How much money should we spend on looking for something that we hope is not there? What treatments are effective? Are they available where? Even on small scale local, but also country to country, different parts of the world. Prevention, what's in place? What could be in place? Pushback on mandates and lockdowns, and should we have lockdowns roll up, and when should they go down? What about children in schools? Vaccination, the coverage, where, what programs are available, equipment, and so on. So there's a lot of constraints in terms of where you can put your effort. Should you move the equipment around? Should you concentrate on vaccination or prevention or treatment? The multiple data types and sources, you all have seen, there's been a plethora of published reports and preprints on you making use of different types of data, multiple sources of the same type of data. So maybe surveillance data from different countries. They may be of different types and different scales of accuracy. We have privacy and confidentiality issues on information on individuals. In outbreak settings, the discussion is necessary and appropriate, but once you decide something's appropriate, can you put the genie back in the bottle after the outbreak has subsided? There's syndromic data or data about your symptoms. There's models and projections, maybe additional proxies, and the accuracy and reliability is variable across these different sources. So how do we live in this world and try to get quick, accurate, and precise answers? It's a very complicated optimization problem. As in the introduction, I pointed out I was trained in operations research, so you can think of this sort of engineering problem of multiple constraints. You have a limited budget, you have limited time, and we have limited testing available, or at least we did in the beginning. As with the rapid tests, there's more availability, but how do we make good use of that? What's available? Look where the cases are, check where it's going, check places of high consequence. So rather than find the precise mathematical answer, you tend to focus on past experience. Where have we seen it before? Now, our past experience only goes back a year and a half, a little over that, but we're still learning and being able to adjust. And we want to watch where it would be particularly bad, say high population density, low vaccine coverage areas. Maybe we want to focus our surveillance there. Maybe we want to go on, this area has had low prevalence for a while and we're worried that it's next to high prevalence area. And then if there's resources left, you may distribute some of your information. If you think of our data sources as like sensors that we're putting out in the world, we want to concentrate them where we'll get the most information, and we also want to concentrate them on places where it would be, we don't want to miss the beginning. Now, I think one of the things we have to remember whenever we pull data together and start looking for patterns, whether those are spatial patterns or temporal patterns or statistical patterns or epidemiologic patterns, is the pattern in any given data set is a combination of patterns in disease risk. That's what we're really trying to see. But it's also patterns of the population at risk. So we may see more cases faster in higher population density. We may see more cases in the populations that have lower local vaccination coverage. And there's also patterns in our data collection system. If we have really good, rapid, accurate tests in some part of our study area and slower and not so accurate in some other places, that's going to be a filter that's going to change the true map into what shows up in the data. So it's not just grabbing everything we can, because some of the material that we're getting is presenting a filtered and biased picture. And also patterns occur at multiple scales. There's the scale of transmission, how often people are near each other and what numbers and how does that play into transmission. There's the scale of observation. If we're only seeing the total number of cases in a given time or an area, that often doesn't give us enough to identify the biological parameters we're interested in. So for instance, in SIR models, which are a really backbone for a lot of our modeling settings, we often try to fit those to data that are just numbers of cases per week or over a period of time. But then you can't tell the difference between something that's very transmissible, but there's not a lot of contacts, or something where it's not very transmissible, but there's a lot of contacts. So you need this extra information, and how you're collecting it can limit what you're able to see. We have also the scales of prevention, treatment, vaccination. How are these changed? Again, these will filter the map of what's really going on, but this is the intervention that we're trying to create a filter to change the true pattern in disease risk. This is a picture I use a lot too. If every one of these is a map on a disease ecology setting, if you have host distribution, this might be a zoonotic disease with spread by vectors. Where are, so in the in a Lyme disease setting or lymphatic filariasis, where are the mosquitoes? Where are the people? Where are the infected mosquitoes? And that all interacts dynamically with landscape and climate. This is the kind of mathematical modeling we like to do, like the physicists like to work on. And they all work together to generate the map we really want. Where are the cases right now? Now, if a person is infected here, but the doctors or healthcare workers they visit are not used to seeing this disease, and they only have a small chance of showing up in a diagnosis case, of their case being diagnosed, that filter is another map here. And now the map of diagnosed cases is thinned out in some places. And sometimes we've seen, overzealous diagnosis, so it may be overinflated in others. So each of the individual cases has a chance to get into this map, and that chance is not the same everywhere. Similarly, once diagnosed, they need to be reported to kind of get in the public health data system. That can be another map. Local health data people here might be really fast. Over here, it might be slow. So at any given point in time on public health, we often start with this map on the right and try to work our way back to the truth. And the modelers tend to work here and build forward. And then we have to model how it shows up in the data to line it up. So I will often focus on different parts of this whole system for different research projects. That's not a new idea. This is actually data from London from the 1854 cholera outbreak that we're familiar with John Snow's maps. Here's your epidemic curve of cholera and diarrheal diseases, these two histograms here. And then on the background are a lot of climate or weather variables, rain and humidity and all of that. And we're still doing this. We're trying to find the magic recipe of indicators that triggers that should give us good prediction of the outbreak. So I like showing these examples because we're still working on some of the same problems. We just have more lines on the map. So is it ultimately futile to try to understand the process from a pattern in the data? So like processes may not yield a unique pattern if you have a stochastic system or you have chaos in your dynamic system. And a pattern may not reveal a unique process without additional information. So this identifiability. So both of these are challenging questions. We like to look at patterns in the data and see what processes in the mathematics show up in both directions. And it's not really, can you get a unique answer? But my question is, do I know more now? Do I learn more than I already know after considering this? And that additional information is important. What other data would we need? How can that help? So there's all kinds of data sources we've seen for COVID, syndromic surveillance about pharmacy sales, mobility data from Facebook, social media, Twitter posts, Internet of Things. There's Bluetooth thermometers that every time you take your temperature, it's being sent into the overall database. Google Flu Trends did a fair amount of modeling of flu outbreaks, but then kind of went off the rails at one point. And there's a nice summary in Science in 2014 about we can't just rely on patterns in the data and throw away everything we know about a disease. So I think we're learning that with COVID as well. Location data from phones and mobile devices. How do we link this with clinical data? What kind of privacy? There's a lot of interesting data science of working with private data and access. So good analyses are really technological marvels, fantastic data, nice math, really interesting inferences. But I think great analyses have to have that humility left of, I didn't answer exactly the question, but how close did I get? Is it better to be exactly wrong or approximately correct? John Tukey, the famous statistician, notes an approximate answer to the right question is better than a precise answer to the wrong question. And I think this is important in this context because sometimes the question we're dealing with changes as we grab data. And if we're not thinking about it, we might miss that we answered a different question. So what's best? The rest of the time here, I want to talk about like, who should we test? This was something we were thinking about early in the pandemic, and with vaccination, sometimes testing has sort of slipped away. But if we want to be monitoring for new outbreaks and new variants, how should we do that? So whom should we test? What do we want to learn? If we'd like to know the current incidence and prevalence, we might want to do some kind of random sampling. That's sort of a traditional surveillance setting. If we want to know where are the most severe cases, we might want to focus on contact tracing. And it might make a difference between tests being cheap, fast, and available, as we're seeing now, or expensive, slow, and rare. So there's you can only go so fast on contact tracing. It takes a lot of effort. So if we have a good test, should we just test everyone? And any of you who've taken a probability class where you probably had a homework problem on Bayes' theorem that was like this. Like, what we want is that a test that's good for diagnosing an individual should be good for diagnosing a population, but what happens can often be a surprise. And you may have seen media reports like this. I saw multiple over the course of the year say, tests have high sensitivity and specificity, but more than half of the positive results are false positives. And then so they've got sensitivities 1%, specificities another percentage, and the positive predictive value or false positive rate is a third percentage in one sentence. 3 percentages of 3 different populations, but since they're all percentages, people want 95 and 99 to mean something in the 90s, and to have it less than 50 is just cognitive dissonance. So there has to be some intuition here, and I thought I can write something about this, and then it took us a year to do so. So there's multiple terms and concepts defining the performance of diagnostic tests. People are more familiar with these now. Sensitivity, specificity show up all the time in reports about COVID. So what can help? I want to think about it this way again. So what question do each of these answer? And they answer different questions. They're proportions of different populations. Sensitivity is how often is the test correct for people who have the disease? So what's the probability a test is positive given to an individual, diseased individual? Specificity is how often is the test correct for people who don't have the disease? That conditions on a different background. These aren't fractions of the same population. How do we measure them? Is this what we want to know? If I'm a patient and told my test is positive, I want to know how often does a positive test mean I actually have the disease? So I'm reversing the conditioning, and Bayes' theorem is how we do that mathematically. So I'm not really doing Bayes, I'll jump ahead a little bit here. Bayes' theorem is how to swap what you're conditioning on, what you know, and what you're wondering. So the quintessential Bayes' theorem homework problem, and many of you probably have done this, is suppose you have a diagnostic test for a particular disease with high sensitivity and high specificity. Should we test everyone? And it's kind of a trick question, but in the olden days, in the 1980s, when I received it as a student, it was suppose we have a new HIV test. You can think of cancer screening. Should all women over the age of 40 receive mammogram screening for breast cancer, or men over 40 receive? Should we drop the minimum age to 40 or even younger? Should all travelers be screened for weapons at the airport? There's false positives all the time. You've seen the metal detectors go off, but they're testing everybody. And then some of the travel restrictions should be applied to all non-citizens. So it's sort of this, let's test everyone. And we hope that good sensitivity, good specificity would correspond to this. So Tal Levi and I tried to write how to think about Bayes' theorem without any equations. And this appeared in Harvard Data Science Review. And it took us a year to get it right. And this is our picture with stick figures. So I want to look at the key at the bottom. A person with a red head actually has the disease. A person with a blue head does not. A person holding, what they're holding in their hand is the piece of paper with their test result. If the test is red, they have a positive test. If they have a blue test, then it's a negative test. And if the head and the paper match, that's good. That's the right answer. A false alarm is when they don't have the disease, but the test says they do. And a false negative is when they do have the disease, but their test is negative. If we group people by whether they actually have the disease, all the red heads and all the blue heads, And we use sensitivity of 80 and 90%, which aren't real for COVID. It just works better if I only have 10 people. So I have this sensitivity 80%, 8 out of 10 redheads get red tests. 9 out of 10 blue heads get blue tests. If I take these same 20 people, so half the people in my study have the disease. If I take them and rearrange them by their test results, then I have all the red tests here, and I only have one false positive. And I can check my positive predictive value, 8 out of the 9 people, current healthy people. You can also figure out the false discovery rate. How many of your discoveries, positive tests, were false? 1. So these two add to 100%. Now the proportion of false negatives is now I have all the blue tests and the redheads. So I've taken the same 20 people and I just rearranged them by this, but my prevalence of disease was 50-50. So there's not a whole lot of difference between my predictive value and the others. But if the prevalence is different, now I've got 20%, only 20% of the people tested actually have red heads. Then when I reorganize them and put all the negative tests up here, positive predictive value and proportion of false positives change. So we were trying to show what happens as you reorganize, and you can see that these are proportions of different things. Another thing to see how you get false positives is to have 50-50 here. I've got 99% and 95% on the sensitivity and specificity. If you start testing more and more people without the disease, pretty soon your positive tests, which are wrong, outnumber the positive tests that are right. So applying a good test in a low prevalence setting will result in a lot of false positives. There is some intuition to that, but that's what we were trying to show and not just give an equation of how it flips around. So it basically comes down to small percentages of really big numbers. If you have a small chance of getting a false positive, but you test millions of people, you'll get a fair number of false results. And so we should use this in designing our studies, thinking forward, but we all seem to have completely forgotten this homework problem over the course of the pandemic. And that finding a needle in a haystack depends on the relative size of the needle in the haystack. It's an easy problem in a small haystack into the big needle, and it's a harder problem the other way around. I think an important part of our quantitative work on infectious disease is to build intuition and expectations, not only among our research community, but among the people we're talking to, the policymakers and the general public. So this is a whirlwind tour. Public health surveillance involves multiple goals, multiple constraints, multiple data types. So it's hard to find one best way to do this, and rather we should come up with best concepts. And they should be informed by our models, and they should be informed by our data. Infectious disease surveillance adds additional complications, particularly when it comes into time, because you're infected for a short amount of time, and we're trying to do the surveillance to interrupt that cycle. Patterns in surveillance data are a combination of patterns of the disease, patterns of the population at risk and risk factors, and population of the data, patterns in the data collection. And diagnostic testing often has multiple goals. At the individual level, we have a goal. Am I infected? Can I go to work? And how many people, what fraction of the population? And has multiple measures of performance, which are all different dimensions of an overall performance system. So I'll just end with a quote from Ralph Waldo Emerson that nature's dice are always loaded, and her heaps and rubbish are concealed sure and useful results, but they're not always. We need to think hard about finding them, and we need to be careful in communicating them. So thank you very much for your time.
Thank you, Professor Lance Waller, for the wonderful talk. Actually, I'm reminded of this saying, research is not about giving a solution to the problem. It's about asking the right question.
Yeah.
So especially when we are going through your, when I was listening to your presentation, the first half of the presentation were questions, asking the right questions. So much of research is being done, but asking the right questions actually led to the correct answers. So research is, maybe I would say 70% always asking the right question, then answering the right question.
Right.
Thank you.
Yeah. So it's very hard to identify the question we should answer, but we often are very quick to answer the questions we can. And so that's where the humility comes in, that I have a certain set of skills. I can answer a particular type of question, but I need to be honest about, am I answering the question that is being asked, or am I answering the question that's easy for me to do, or, that from my experience, I know how to do. And it's hard to admit when you don't know how quite to get the right answer.
Yes. Yes, sir.
Well, is there any question, Dr. Vamsidasi, Dasu?
No, sir. From the participants, we don't have any questions.
Yeah. So just a quick thing, Dr. Lance Waller has an incredible background also in drugs and alcohol drinking, and he has done tremendous amount of work, and literature is full of his papers on that. So if substance abuse, we had a conference in Banff, Canada, and it was with the field scientists as well as mathematical modelers, and Dr. Waller was also a co-organizer in that. So I think we had an incredible work that we talked about there. So not only that he works in this particular area of disease related and COVID related, but also in other related. One thing also I want to point out is his paper that he just cited in his slides also, which was in Harvard Reviews, I think, right?
Yes.
Yeah, Harvard Data Science Review. Mm-hmm.
It is must-read paper,
Thanks.
and it is beautifully explained. It is beautifully explained. I loved it, it's very minute concept, but it is very involved concept also about sensitivity, specificity, and without that, we model these testing in our models. How effectively we are capturing in these mathematical model, I don't know, but I think those understanding is very key to capturing the right way of the models also. So just a couple
we
of
have to
points
thank
also.
the editorial process. They have a very detailed, multiple experts, and a very careful writing review. So the paper is much, much better than the first version we submitted. So,
Yeah.
as is often the case, the referee process may be challenging, but it does play a role in quality.
So, Dr. Waller, I did a project recently on comparing the point of care rapid testing versus PCR testing.
Mm-hmm.
And this was very key element, how you capture the sensitivity, because what happens is you take these rapid testing, which is 30 minutes, but then if you are false, true negative, the false, if you are not a case, if you are a case and the test doesn't recognize you as a test, then you think that that I'm not having a disease, and you go in the community and spread the infection. So there's a huge outbreak comes from you as a super spreader kind of.
Mm-hmm.
But your behavior changes also in the population. So it is a key element actually that we find it is very key to the modeling.
Yeah, and the mathematics works out assuming everyone acts the same way, but if somebody gets a positive, sometimes they keep taking a rapid test until they get a negative, and you have to build that into the math too.
Yeah. So how is this, just one more question actually, how is people capturing these, there are different tests like antigen versus antibody based, what is the benefit of when to use each one of them, and these things are very key there.
Yeah, and I think the design questions are really interesting. I will tell you, how Emory does their rapid tests for students, which they don't do as much now with a vaccine. We have a vaccination requirement, but the quick one is not it's a saliva-based one, which isn't so, but they use that as a screening to take the one, how many of, because they were testing all the students, who should they do the more expensive, more accurate tests on? And this is similar to cancer screening too. So mammography and PSA testing are not particularly strong tests on their own, but they're quick. And if you're positive on that, they don't give you a diagnosis of cancer. Now they'll do a biopsy. And so when you think about the mathematics of this, it's not just sensitivity and specificity of a one-time event. It's the combination of the protocol. So you can really get, it's fascinating to me because it's one equation, but the pieces are are challenging, and to try to model the screening system accurately is a challenge. And the cancer one, there's a fair amount of time between you receiving a positive screening test and your biopsy. That's a very stressful, unpleasant period for someone who's had a positive screen and then is told negative, and you may lose them to future screening because they don't want to go through it again. They might say none of this means anything, and we have to think about those implications too.
It's true. Thank you very much, Dr. Waller.
Yeah,
It's
thank you.
amazing talk.
Yeah, thank you. Once again, we thank Dr. Lance Waller. So, thank you all the participants. We will close the day one evening session. And tomorrow morning at 9 a.m. sharp, we'll meet for the day two proceedings for the Indo-US conference too. Thank you all once again. Good night and good
Thank you
afternoon
very much.
for the
Good
US
evening.
speakers. Thank you. Thank you, sir. Thank you, Dr. Anuj.